Hello everyone, I'm early stage researcher number 6 in the Oberon project, and today I'm going to give you more information about this project and my role in it. This project is funded by European Union's Horizon 2020 Research and Innovation Program under the Marie Curie grant. Uh, Oberon is a European collaboration that brought together a group of scientists that work on the field of optics and biomechanics of eye and covers a broad range of disciplines and skills. This is an overview of research program. It consists of different work packages 1 to 7, and you see how they are interrelated. As ESR6, I'm considered as in the work package 2 in crystalline lens optics. This graph also shows the interconnection of the thesis of ESRs. My work as ESR6 is related to work of, for example, ESR5, 7, 1, and 10. So what is my role exactly as ESR6? I'm studying optical change of presbyopic eyes in response to accommodative stimuli. Let me introduce the basic of my work. Any optical system has three elements: object, optical part, and image. For example, you can have a scene as object. If you have a photographic camera, it has optical parts which form the image on CCD in this case. But our eye also is an optical system. As you see, it has different parts that form the image on retina. Cornea is transparent and contributes 70% of the refracting power in the eye. The iris is the color part of the eye that controls the amount of light. Crystalline lens is a transparent elliptic structure that focuses light rays on retina. And capsule of the lens is connected to ciliary body via zonules, as you see. Accommodation is the process that the optical power of eyes change to maintain clear image of the object at different distances. The process of accommodation involves contraction of ciliary muscle, which is attached to capsule of the lens via anterior zonules. As the muscles move forward and inward during contraction, these zonules relax. This is shown in this picture. When ciliary muscles are relaxed, zonular fibers are tightened. For accommodating, ciliary muscles contract, so zonular fibers relax and lens power increases. Now we can visualize the accommodation process in this video and see how lens shape changes with distance of object. After understanding the accommodation, we can get familiar with another term, presbyopia, which is gradual loss of the eye accommodation due to aging, so lens cannot change its shape, and senior people have difficulty for seeing near objects, and they use positive lens for reading. Presbyopia starts around age 40, and around age 50 we can see eyes lose accommodation ability completely. But still small changes in lens position and geometry are possible. Lens may drop downward due to gravity, which affects third order aberration. What is aberration? We told that optical systems have three elements because none of optical systems are perfect. So any distortion of image from the ideal optical image is called aberration. This distortion can be explained mathematically using Zernike polynomial. These polynomials have different terms and some of them have specific names, and aberrations can be decomposed as a sum of these terms. So lower order aberration, which are defocus and astigmatism, can be corrected by eyeglass or contact lens, but not more. In presbyopia, lens cortex may keep also some elasticity and move around the nucleus, changing equatorial diameter. Subjective effects of all of these are visual fatigue, blurred vision, and diplopia. So we developed first experiment to study the visual tasks using an iPad and e-ink reader that we're choosing to put in a color assessment cabinet. The subject here were fixed on chin rest 50 centimeter away from screen, and we had two lighting conditions: high ambient illuminance with setting maximum luminance on iPad and e-ink reader, and low ambient luminance with setting minimum luminance on devices. We have we had 24 young healthy subjects. We asked them to read the text for 5 minutes. Eye movement during reading task was monitored and recorded using eye tracker, and potential optical change were assessed by comparing aberrometry measurements before and after study. This is an example of aberration change after reading task. Changing of wavefront errors can be seen here. We analyzed the results of aberrometry and eye tracker. Eye tracker data revealed a higher number of saccadic eye movements under minimum luminance. The length of saccades was rather higher for minimum luminance condition for both devices. Pupil diameter and blinking rate increased significantly for low lighting condition. This implies more visual discomfort, which was consistent with subjective opinions. We also saw that higher order aberration varied after finishing reading, although differences were not significant compared to initial stage, showing visual recovery of ocular system because our subjects were young. In our second experiment, we used 13 healthy volunteers that were developing the presbyopia. We asked them to read text, this time on a laptop, in the same with the similar highlighting and low lighting conditions. Participants rested their chin and forehead, and the distance to the screen was 20 centimeter to 40 centimeter. This distance was chosen in a way that made the subject's eye have maximum accommodating effort, where the text to read started to blur. The subject were asked to read text for 20 minutes this time instead of 5 minutes under two lighting conditions. So aberrometry measurements were performed using ILX aberrometer again before reading, right after doing the activity, 5 minutes after activity, and 10 minutes after activity this time. Measurements were made in triplicated at each time point and then averaged for subsequent analysis. Aberrometric data of each eye was rescaled to the smallest value of maximum round pupil as recorded during each measurement. The measurements revealed that immediately after engaging in visual tasks, there is an increase in defocus aberration, but this value returns to the baseline level in 48% of the eyes during subsequent measurements. As for C40 coefficient, which represents primary spherical aberration, it initially decreases immediately following the task and then increases in 36% of the eyes. Regarding vertical coma, a pattern of increase followed by decrease is observed in 58% of the eyes. This phenomena can potentially explained by downward drop of the crystalline lens during accommodation caused by gravity, leading to change in higher order aberration, as we mentioned. Total RMS of aberration increased after finishing the reading task and then decreased in the subsequent measurements. RMS of higher order aberration, which include chromatic and spherical aberration, showed a similar trend of change. However, RMS of spherical aberration decreased after the task and recovered in 5 and 10 minutes aberrometric measurements. As expected, measuring higher order aberration right after near vision tasks show more increase in low lighting condition due to pupil enlargement than highlighting when we see a similar trend. In the third experiment, we tried to see the effect of small angle misalignment on ocular wavefront Zernike coefficient. We asked 10 volunteers to look at different points of fixation E-letter of aberrometer. The wavefront aberrations of their eyes were recorded 3 times and averaged for analysis, and the focus was compensated with help of Badal system of aberrometer, so object could observe fixation E-letter clearly without wearing vision aids or accommodating. Analysis of these results showed that C40 coefficient increased in 50% of the eyes when subjecting their fixation gaze from zero point 0.1 to zero point 0.2. Moreover, C3-1 coefficient indicated decreasing trend in 55% of the eyes under the same condition. The change resulted from the altering the fixation gaze are in the range of change observed after a near vision task in our second experiment, and might be a confounder when trying to detect such small changes in the wavefront error. According to the results of the last two experiments, it was found that after sustained visual tasks, different lighting conditions, small changes in Zernike coefficient can be detected, demonstrating some predominant patterns. However, considering that the fixation target uses an extended object, here E letter, this alignment might contribute to random changes, which could make it difficult to detect minor anatomical or physiological change in the human eye over time. So we suggest instead of using this kind of fixation when we use, for example, Maltese cross. And our next step for experiment is trying to study using ophthalmic electrophysiology, which is a series of tests that measures how different parts of eyes and vision systems respond to light. Visual evoked potential, or VEP, is a minute neural discharge that occurs in human visual cortex in response to patterns or other stimulations of the retina. Many factors may affect VEP testing. Aberration is one of the most important influential factors because it directly degrades the retinal image quality of the pattern stimulus. We are going to investigate the effect of higher order aberrations on VEPs. So these are some of my scientific reports as poster or paper. Thank you for watching this video. I hope you enjoyed it.